part two of this process of getting a gene to turn into a protein is called translation. So now what we're doing is we're taking the information that's stored in this new messenger RNA molecule that's been prepared from a DNA molecule in the nucleus. We're bringing it to the ribosome, and now we're going to translate that information into an actual protein. All right, so last time I wrote stage 1, and now you want me to write stage 2 for this one. So there you go. Stage 2 of this entire process. All right, so now we have to look at a couple of other forms of RNA in a little more detail. The first thing I want to talk about is this transfer RNA. Transfer RNA, or tRNA for short. That's usually how we write the transfer RNA. Transfer RNA is an interesting little bundle of RNA. Looks something like this. What it does is it rolls over on itself, and it creates. So let me just draw the base sequence. It rolls over, it kind of bundles up like this, and it creates this little bubble on one side. And then it bundles down like this, and it creates another little bubble on the bottom. And then it creates another little bubble on this side. And then it just sort of goes like that. So this is the 3' end of the molecule, the 3' end. And this over here is the 5' end. And it's sort of folded around on itself. And the reason it's folded on itself is because there is some attraction between the bases on each side of this molecule. So in these regions that I'm drawing here, where it's sort of stuck together, the bases are attracting to each other and making hydrogen bonds, right? Very much in the same way that the 2 strands of DNA are held together. But these little bubbles on either side are formed, and these bubbles have sequences that don't really bind to each other very well. And the bottom one here is the one that's important. So what they would have here is a sequence of bases. And right on the bottom, I'm going to write 3 of them. A, G. Let's use that example. Now that is called an anticodon. Because it's the exact opposite of a messenger RNA codon. So by opposite, what I mean is the RNA codon that would attach to would be UUC. Remember how U's go with A's, like T's do, and G's and C's go together. Well, this anticodon is very important because it's sort of like the sticking spot. This transfer RNA is able to bond with a piece of messenger RNA at that particular spot, at that active site. Now, behind the RNA, I guess I'm going to have to erase. Do you have room up here to write? No? Okay, then let's just draw another picture. We'll draw it again. Behind the transfer RNA, and I'm just going to draw a box here. I'm going to put AA in here, which stands for Alcoholics Anonymous, or amino acids. Amino acids, right? So I'll put it ... So this is a particular amino acid. Maybe we'll just put 1 here. There's no point in it. And this amino acid attaches to the end, that little tail. So now I'm going to draw that RNA guy again. So he's attached here. And then he's got this little bubble here, this little bubble down here, little bubble over here, like that. He's got some bonds, hydrogen bonds here between all these bases that are here. These are relatively small molecules. They probably have somewhere around 30, 40, maybe 70 bases all told. And so this is a hydrogen bond or a light weak bond to an amino acid. So they serve like a taxi. And then, of course, we have our codon down here, which was, what was it, AAG? And so, if I draw a piece of messenger RNA down below here, if this messenger RNA happened to have a sequence a sequence down on it that was UUC, then there would be potential for a weak, a weak hydrogen bond or a link here. So on the messenger RNA, the UUC is the codon, and you'll remember the different codons. If we go back to that chart in our book, UUC, and we find that codon, where would it be? UUC stands for phenylalanine. So that means that this amino acid, because this particular transfer RNA has a very specific base order, this link up here is only going to be able to hold phenylalanine. So in this case, the amino acid that would be pulled here is phenylalanine. And that's because this code right here is the code for phenylalanine. And then the anticode, of course, is what matches with it. So we could just as easily make a chart of all the anticodons. But by convention, we've decided to use the messenger RNA as the definer of the codon sequence. We could say that AAG pulls phenylalanine, right? Because it does. That's the taxi's license number that pulls this amino acid. But instead, we've decided that the messenger RNA is what we would call the codon. So the green letters are the anticodon. So now you can kind of imagine what's happening here. In a fairly complex process, which involves the ribosome, okay, so now let's draw the whole thing. So let's just draw here on what? Phenylalanine. Phenylalanine, that happens to be the one that corresponds to that codon. Okay, so now let's sort of draw the entire process sort of happening. So what we have is a large ribosome. So let me do that in a big purpley color. Like this. Big, big. The ribosome has on it a sort of an active site where the messenger RNA can be held in place. So in blue, I'm going to draw a piece of the messenger RNA like this. mRNA. And the messenger RNA would have letters on it. So let me write some letters in blue. And I'm going to space them out a bit just because I want to see the I want you to see the codons. They wouldn't necessarily be spaced out, but so let's put U, GG here. And then UUU, and then GGC. So the spacing isn't really there, but just to keep the picture from getting too complicated, that's a GGC, that second one. I'm just spacing out the codons so you can see them, right? But you understand that they would be right next to each other. Okay. G, that's worse, GGC. And this would be all lined along here with the codons. And so basically what happens then is a piece of transfer RNA, which I'll put in red, sort of comes along here. Yep. I'm using red for the transfer RNA. And over here, I'm going to put one sort of coming in. He's not quite there yet. He's on his way. Oh, he went wonky on me. That's okay. And then I'll use another color, maybe green, for the amino acids. So this amino acid that is attached here, I'll put it in a green box. This will be a different one, so I'll make it a triangle. And this would be a different one, so I'll make it a circle. These are all different amino acids with different transfer RNA molecules. Each transfer RNA has only one specific protein or one specific amino acid that it carries. So if you look at the anticodon on this, you would have to see here an A and a C and a C. And on here, there'd have to be an AAA. And on this one, coming in, there would be a CCG. Those are the anticodons that would form weak bonds and line up. So this one is moving into place. And then what happens, of course, is as they move into place, you could see all these amino acids being sort of shuttled in behind them, lining up. Okay, well, the letters are going to vary. So whatever letters there are on the messenger RNA, there's a whole bunch of different letter combinations, but each combination is a 3-letter codon. Remember? We talked about that. And so the transfer RNAs have an anticodon that matches it. It's the complement to it. So when they come nearby, they can easily attach or sort of bind. And if we look up those 3 blue codons in the chart, right, the first one is UGG, and so UGG is tryptophan, I think. Yeah. So this green one right here, his actual name is tryptophan. Tryptophan, because of the UGG on the blue, right? And then the next amino acid, the triangle one, if we look up UUU, we've already seen that one. I think it was phenylalanine. And then the last one, which is a GGC, would be glycine. And so we are actually putting the amino acids together. Once the amino acids are lined up, then enzymes can catalyze the peptide bonds between them. And the amino acids can then be linked together by a peptide bond. Tryptophan, phenylalanine. Yeah, you can't really see that, can you? From way back there. Tryptophan, 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 phenylalanine, and glycine. And then I've written peptide bond. I should have put peptide bond in a different color because it's not another amino acid. So let me just change that just for fun. Let's call it, put it in what, maybe this brown color here. I don't know if that's going to work. Is it going to work? Yeah. So right here, we'll put peptide bond formed by enzymes. Now, all of this is happening on this large purple structure, this ribosome, right? So the ribosome, I'm just going to color a little bit to give it some depth here. So the ribosome is this big structure. And it has active binding sites for all these molecules. It has places where the transfer RNA can shuttle itself in. These other sides to the transfer RNA that don't contain the anticodon but are little sort of spots, they would obviously be interacting with the ribosome and holding themselves in place. So part of that they're active as well, but they don't attach to the messenger RNA at all. So the ribosome has active sites, just like a regular enzyme would, except this is a complex of several protein enzymes and also ribosomal RNA, which is also attached here. The ribosomal RNA serves as places where the messenger RNA can attach, and everything is held in there by these sort of hydrogen bonds. Yes? Yes. So if we were to summarize, the important thing here, it's that the DNA sequence of bases gets transcribed into a messenger RNA sequence. And those blue codons are the actual sequence of the gene. And it's a code or a message that tells the transfer RNAs exactly how to line up. There's only one way they can do it. And when they do, they line up and put a very specific sequence of the green amino acids next to each other, allowing the enzymes to form the peptide bonds. And so the blue letters are the secret code that translates into the sequence of amino acids, which in effect is now a polypeptide chain, which now forms a protein. And the function of that protein is very, very, very tightly linked to the sequence of the amino acids within it, because they all would behave chemically in a different way. And they would form different shapes, right? Different secondary, tertiary, and quaternary structures based on that. And so that is essentially the process of building this polypeptide chain or protein. Okay, so we'll stop this one.